Hi, it's Mr. Anderson, and today I'm going to talk about linked genes. When I teach biology, one of the hardest things to explain is linked genes. And so I'm going to kind of go through this slow and make sure that I hit all the points, and hopefully you can have that aha moment when you finally understand how linked genes work. Most of this is through the work of Thomas Hunt Morgan. He bred fruit flies and kind of took the work of Mendel and then pushed it into modern day. And so this is the early 1900s. And so the people, Thomas Hunt Morgan, the people who worked in his lab, discovered so many things that we now kind of take for granted. First of all, these are fruit flies. Fruit flies are really hard to see with an eye. You have to look at them under a dissecting microscope. But basically, you grow them in a medium like this. They'll lay eggs. You take the eggs out. They'll hatch into larvae, and then the next generation of flies shows up. And so this is what a fruit fly looks like when you could zoom into it. You can actually, if you were to look really closely, you'd see that there are two types of fruit flies here, and they're separated by gender. In other words, this one right here is going to be a male. It kind of has a rounded rear end, and then it also has these things up here on its arms. Those are called sex combs, and the sex combs are a good way to tell the difference between the male. So this would be a male right here, and then you could tell right here, no sex combs, kind of a pointy end, so this is going to be a female right here. And so you could go through and figure out which ones are male and female, but that's not what we're interested in. What we're interested in is linked genes. And so in the fruit fly lab of Morgan's fruit fly lab, basically whenever he found a mutation, then he would breed that mutation until he had a pure amount of that. And so you can see here that this is a fruit fly that's got two mutations. First of all, it's got a color that's black, and then it has these vestigial wings. And so everything else is going to be same between these two fruit flies except the color and then the wings, vestigial wings. They can't fly because they have these little nubbins of a wing. And so basically he was doing work on fruit flies. And so the nomenclature for genetics on fruit flies is pretty simple, but it can be a little scary if you've never seen it before. So basically we take a trait. In this case, this BLBL is going to stand for the black mutation, and then the VG stands for the vestigial wing mutation. And so then you just take those letters and then you add a plus to it for the wild. And so this would be wild type for both of those genes. Okay, so let's say we were going to do a cross between these two. The way you would set it up, and again, if you're not sure on how to do this, make sure you watch the podcast on Punnett squares. But basically, this one can either give the vestigial or the vestigial, or excuse me, the wild or the wild, and this one can give the wild or the wild. So basically, in meiosis, it's only going to be able to produce this one gamete right here, the female. The male over here, since it has two of the mutant genes, it's only going to be able to produce this gamete right here. And so if we were to do a cross between the two, this would be what we get. So we're going to have a hybrid for these two traits. So it's going to be heterozygous, heterozygous. And so you might think to yourself, what's that fly going to look like? Well, if these are recessive genes, then we would expect it to look like a wild type. If these are dominant genes, we'd expect it to look like the mutant type. But if it's a combination of the two, we might have one that's black with normal wings. So let's figure that out. So I'm going to go to the Fruit Fly Lab. So Fruit Fly Lab, this is at sciencecourseware.com, BCISE. And so basically you can do any cross that you could imagine. So I'm going to order those fruit flies. So I'm going to order the flies. Again, the female was of the wild type, so I'm going to add that to the cart. And now I'm going to figure out the male. So the male, again, had a body color that's black, and then it had a wing size that was vestigial. So I built the male. I'm going to buy that, and I'm going to check out. And they already arrived in the mail. So I'm going to throw this cross in the incubator. Remember, it's going to hold on to those ones that I ordered, and I'll come back to that in just a second. So I'm going to throw it in the incubator. And so 2 weeks later, we're going to have that next generation. All right. So now I'm going to pull those out, and we're going to, let's try that again. I'm going to pull those out, and we're going to put it underneath the microscope so we can see what came from that. So throw that under the microscope. I'm going to sort it. Normally you apply like ether to the flies, and then you can separate them with a paintbrush, so you don't want to kill them. They're just asleep because you're going to use them again. So what I got is 2 piles of flies. And so let me look at the female right here. So the female right here is wild for color and wild for wings. And so we know that those 2 mutations are recessive. Now what I'm going to do right now is add this and use it in a new mating. So I'm going to add that, and we'll come back to that in just a second. Let's make sure the male, so the male's over here, those are all same thing, so they're going to be wild as well. And so it looks like those 2 different traits were recessive traits. So let's kind of go back to the keynote for a second. And so what did we get for a fruit fly? We got wild type of those 2. Okay, so that made sense to Thomas Hunt Morgan. This was just Mendelian genetics. And so if he would have stopped there, he wouldn't have learned anything. And so what I'm going to do is take this, the daughter in this trait, and let's cross that with a homozygous mutant for both types. Okay, so now we have a hybrid over here, and then it's essentially a daughter from that cross, and we're going to cross it with the dad in that first cross. I know that's gross, but that's basically what we're going to do. Okay, so what could we get for gametes? Well, this one, again, can only give the BL, or the black mutation, and then the vestigial wings. But this one over here can actually produce 4 different types of gametes, because we could have the wild with vestigial, we could have the wild with wild, we could have the wild with vestigial, we could have the black with wild, and we could have the black with vestigial. So there are essentially 4 different gametes that we could get for that. So let me lay those out here. So we've got that. And if we were to do our Punnett square, these would be the 4 types that we could get. And so in this cross, what would they look like? Well, this right here would be, it's going to be wild, wild. This one right here is going to be wild vestigial wings. This one right here would be black wild, and this one would be black vestigial wings. And so this is what the fruit flies would look like. And so according to Mendelian genetics, we would have a 4x1 Punnett square. And so we would expect, if we bred these, that we'd have 25% of each